Thank you, Jeremy, and thanks to ProImmune for continuing this tradition of this conference. This is my favorite conference of the year. I love it, and thanks for inviting me. I changed my title because I saw I was going after Emily, and I figured I might as well get a little broader than just the ELISpot assay qualification. So my group is responsible for characterizing immune responses to all of Celgene's biologics and CAR T therapies. As part of our mandate. And we were charged with measuring host versus CAR T immune responses, and CAR T was new to me and to Celgene at that time. And so the work that I'm showing you is what happened over the last 1.5 years to characterize this. So just a quick cartoon of what a CAR T looks like. Let me make sure I do this right. Yeah. So this is a patient's T cell that has the CAR construct embedded on its surface. This is expressed by the patient's T cells are transfected with a virus that then allows integration of a CAR genomic gene sequence of the CAR construct into the genomic DNA, and then it's expressed and brought to the surface. The CAR construct is a chimeric antigen receptor, so this is the antigen receptor part of the construct. It is essentially the variable domain of a monoclonal antibody. It's attached to a transmembrane domain and an intracytoplasmic signaling domain. When we are characterizing immunogenicity of a CAR construct, what we are looking for is immunogenicity against this extracellular domain, which I will call ECD in subsequent slides. So, so CAR T, when we're trying to imagine what an immune response to a CAR T might look like, we realize that there could be a humoral response resulting in the formation of anti-CAR antibodies. This might result in binding of the anti-CAR antibodies to the extracellular domain of the CAR construct and hence neutralizing it. But the CAR T cell is still there. It's just neutralized. It cannot bind to its target anymore and therefore not functional. The other immune response that we could envision was a cellular immune response resulting in the development of cytotoxic T cell response against the CAR T cells, so host T cells versus CAR T cells. In this case, we can envision that the CAR T cell numbers actually go down because the CAR T cells are being killed by the cytotoxic T cells. And to capture each of these events, of course, we have PK assays where we're measuring the CAR T cells in two different ways. One is a qPCR-based vector copy number assay, where we're looking at the number of integrated vectors into the T cell genome, and that tells us, as we are measuring it in patient samples over time, that we still have integrated vector-containing T cells, but those T cells may or may not be active T cells. They are there, physically present. So you could imagine if T cell is neutralized by a humoral response, we'll still measure it in the VCN assay because it still exists in circulation. Whereas in this case, you will actually see the T cell numbers going down. So what we are hoping for is to take this immunogenicity data along with efficacy data, along with the PK data from these two different assays, and together try to come up with answers to questions like, will this particular patient benefit from a reinfusion? If we want to reinfuse this patient, do we express an immune response? Do we expect the reinfusion, the expansion of T cells post-second infusion, do we expect it to look exactly the same as the expansion of T cells, CAR T cells post-first infusion, or how does it change? What should be the timing of the reinfusion? Should we immunodeplete these patients before we reinfuse, or is the first immunodepletion sufficient? Do we have memory responses living post the immune depletion that we do? So do the memory T cells get depleted when we do the immune depletion prior to CAR T therapy, or do we still have memory? So lots of questions, and clearly any one piece of data is not going to answer it, so we're just collecting pieces of data from everywhere to put it together to try and get answers to these questions, and the pieces of data, two of them are what these assays are going to provide. So the objective was to measure immunogenicity against CAR Ts. We used two different methods to detect the humoral response. We did an electrochemiluminescence, it's an immunoassay, based assay to detect anti-CAR antibodies. And to detect the cellular immune response, we went with a classic ELISpot-based assay, and this was done with the help of ProImmune. And I'll walk you through some of the challenges that we faced with each of these two assays. Yeah, so there we go, the challenges. So we had very little experience. I'm not an immunologist. I come from a PK. I'm a PK scientist, so I was doing PK of biologics and PK along with the immunogenicity gave the full picture. So I got into immunogenicity and then now CAR T. So it was a little bit of a challenge, and ProImmune was my immunogenicity 101 classes. I got it from ProImmune. But anyway, the challenges we faced. First, generation of relevant and specific positive control for the CD8 plus T cell assay. So we needed a positive control in order to be able to characterize our assay, and that was the first challenge. Reducing interference from CD4 plus T cells. So of course we can do sorting, but we did not have enough, we will not be able to get enough sample from these patients to be able to sort and then do an ELISpot. So we wanted to be able to reduce interference, addressing the constraints of a lymphodepleted population. So the way the CAR T therapy works is we lymphodeplete the patients right before we infuse them with the CAR Ts. The PBMC yields are low. It takes time for the PBMCs to come back, and so we were not confident that we will get enough PBMCs to do everything that we wanted to do. Also optimizing sample collections to strike a balance between feasibility and probability of generating meaningful data. So this sentence actually holds a lot because we needed PBMCs. We know the yields are going to be less. We know we want a large sample volume. There's limitations on how much we can collect from these patients. And we wanted to make sure that with all those limitations, the data that we get is meaningful and able to support interpretation of in the context of everything else that I just talked about in the beginning. And of course, to develop a strategy to deal with poor quality or insufficient PBMCs. So all of these issues were dealt with as we went through this development. So I'm going to talk about the humoral response first, a few slides on that. Initially, as we were discussing this internally, the first humoral response assays were done using CAR T cells as a reagent. So we took donor PBMCs, turned them into CAR Ts, and used that as a reagent to capture the anti-drug antibodies, which would then bind to the ECD of the CAR Ts, and then to detect this. It was expensive, as you can imagine. We had to make reagent CAR Ts. There was reproducibility became a big issue because there was lot-to-lot variability, enough to throw these assays off. And other problems was high background, especially we were trying to deal with multiple myeloma patient samples, or rheumatoid arthritis patient samples are also a problem. So we had high background, low sensitivity because of the high background. And the positive control that we were using was not the best, so we had to go back and redo this, and poor reproducibility. So we changed it around to going back to our biologics. We do this all the time for a routine biologic, so we said let's just not deal with the CAR T as a reagent. So we produced the extracellular domain of the CAR T as a recombinant protein, used that as both the capture and the detection reagent. The capture reagent is not labeled, the detection reagent is labeled, and used a classic bridging ELISA-based assay to detect anti-drug antibody responses. And with this, we were able to overcome pretty much every problem that we faced. You can see here, we also generated a polyclonal affinity purified rabbit anti-ECD positive control antibody. which was of higher quality than what we were dealing with before. And together we were able to get a highly selective, highly sensitive assay. So the sensitivity was in single-digit nanogram ADA, and it was selective in disease state serum, which is what we were hoping for. So on to cell-mediated immunogenicity. So this is the work that we did with ProImmune. Where we wanted an assay to detect CAR-specific cytotoxic T cells in PBMC samples from dosed patients. And we initially thought what would be the best approach. We considered an interferon gamma ELISpot, we thought we had the most experience with that, and it was a medium throughput and a sensitive assay. We also considered tetramer-based approaches and decided it was not practical because we wanted to deploy this assay for clinical trial samples across multiple clinical trials. We were going to have all sorts of patients with HLA background, and we did not want patient-specific reagents or the HLA restriction to be a constraint. And also we looked at some granzyme B and perforin detection. Our therapeutic here is a T cell itself. We had false positives and poor reproducibility. So decided to stick to the ELISpot. The challenges, again, are low PBMC yield due to lymphodepleted patients, and identifying the timing of maximal anticipated response so that we don't waste our sample volume on poorly timed sample collection. And also the logistics, we needed to work with frozen PBMCs. Shipping fresh PBMCs from every single patient in a multi-center trial, global trial, was not going to be a possibility, and of course to generate positive controls. So I'm not going to go through the ELISpot. Everyone's done it a lot more elegantly than this picture I put together. But we took ProImmune's already available interferon gamma ELISpot assay and optimized it for our CAR ECD. Of course, the assay was optimized to work on frozen PBMCs. We took ProImmune's recommendation on how to collect the samples, how to process them, and freeze them and ship them. And it's not as easy as it sounds because you are working with central labs. They don't want to change anything. Their people are trained on their SOPs. If yours differs, half the people follow the SOP, half the people just do what they're used to doing. And it's not as easy as one bullet, but we are getting there. We used 9-mer peptides as a way to not invoke responses from the CD4 plus cells. As I said, cell sorting was not an option. We didn't have enough cells, so we figured using 9-mers would predominantly restrict our responses to CD8 plus T cells. And we tried working with lower cell counts, and I'll share the data with you. And yeah. So I think we can skip that. Yeah, and this is the point I want to make. So the pool consisted of 9-mer peptides, and we wanted to cover the entire ECD, including the variable domain, the spacer region, and also the first few amino acids of the transmembrane domain as a just in case. We ended up with 311 peptides to cover it as a 9-mer peptides within 1 amino acid offset. So we have 311 peptides. We can't make smaller pools, subpools with these because that would mean we would need that many more PBMCs. We have PBMCs to do one shot at best. So we needed the entire 311 peptides as a single pool. Scientifically, I can see 100 caveats from this, but that's all we had to work with. Subpooling and running 6 or 7 ELISpots with 50 peptide pools was not an option, practically speaking. And so we did go with a pool of 311 peptides. So controls, the negative control is an unstimulated control, exactly as ProImmune had it. Positive controls, again, exactly as they had it, PHA and CEF. So PHA is essentially a control for PBMC quality. If the PBMC quality is good, the PHA definitely needs to show a good response. If this doesn't work, you don't have good PBMCs, period. The CEF peptide pool was a less sensitive control, but this was interesting. We had an option of completely leaving this out, but I thought this was interesting because the CEF peptide pool works because the patients are expected to be exposed to these kinds of peptides during the course of their lifetime. They have memory response, and so when you add this peptide pool, they should produce interferon gamma. So the question was, in our lymphodepleting regimen, what do we deplete? And how robust is this memory response in these patients? So it was a little bit of an extra question that we hope to answer if we could with this CEF control. And a CAR T specific control. This is an addition to our assay. So these were all part of the standard interferon gamma ELISA. So CAR T specific control. So what we did is to generate our own positive control, we purchased donor PBMCs, stimulated them with our pool of 311 peptides against the CAR ECD, allowed these T cells to grow and expand, and then froze down those expanded T cells as our positive control. And they are frozen down as single-use aliquots, I believe, so we can use them not only during our assay validation to validate with our own pool, but also to use it during study sample analysis as a positive control on every run when we are running study samples, because we hope to use this over multiple years. And maybe we have to synthesize multiple peptide pools to have enough. And this would be a good control to bridge all those years and all those different runs and different peptide pools. And so so you can see here 5 different patients with 3 of them having robust response, and we purified these and pooled them. So lower cell counts, I thought this was interesting. So ideally 250 million cells, is that? No, 250,000 cells per well is what ProImmune recommends. That's the ideal situation. We dialed that down to 200, 150, and 100 and tested it with PHA, CEF, as well as our CAR T peptide pool to make sure that we are functioning. We did go down to 50 in a subsequent program, and we saw that 50 also works, but it went down a little bit. So 100 was up to 100,000 cells. We did not seem to lose signal in this assay. This was good to know in case we had lower yields than what we hoped for. Robust and reproducible, so the assay was run by different users over different days, and we looked at the CV. You can see the controls work beautifully. Negative control doesn't count for CV. Positive controls work very well, and our CAR T peptide pool was also behaving, although the variability is higher with this with our peptide pool. So sample collection time points in the protocol. So this was separate from the functioning of the assay. What would be the best time points to collect? I know this from my biologics. I know exactly when to collect, but for CAR Ts, I had to dig into the literature a little bit and read a lot about transplant medicine. Very interesting stuff, learned a lot. But looks like the peak CD8 plus response is around day 15 to day 80. If you read the entire as much literature as you can across different transplant patients, it seemed to cluster around day 15 to day 80 post-transplant. We decided that day 15 was too early because our population is quite severely lymphodepleted, and we have looked at total PBMC counts in our patients, and really if you collect a day 15 sample, you get nothing but CAR T cells back. So it's very hard to imagine getting good yields at this point. So we collect at months 1, 2, 3, 4, so day 30, 60, 90, 120, post-CAR T infusion, and then we collect 1 sample at the end of the study, and we collect 1 pre-dose during screening. So there are just 6 samples per patient, and these are the 4 critical samples where we hope to see if our patients are positive for CD8 plus T cells against the CAR. And the sample volume is 16 ml of blood. And so preparing for various scenarios, this is where this is what Emily referred to as well, is what do we do if we don't get enough PBMCs? So if we have 3 times 3 million cells, we can do a full set of analysis. We'll have 250 cells per well, triplicate of all analysis. We'll have a PHA positive control, a CEF positive control, a negative control, and our samples. Everything's done if we have this. And then we go down this flow chart, dropping things out. So first thing to go is the CEF test condition. And then we change from triplicate to duplicate for the positive control, reduce the number of cells to 250 to 200 to 150, change everything into duplicates instead of triplicate, and finally drop it down to 100. And we wrote down in our procedure that if we have less than 600,000 viable PBMCs, we will call that sample not determined and not analyze it because we don't have confidence in that. We had to put this down because this is going to be deployed over a long period of time, and we want to make sure when we take this data to the agency that this is done a priori, that we are not picking and choosing which samples we are analyzing and which ones we are giving up on, but here is the decision tree for that. This was another thing where we had a lot of discussion about is what it was is going to be the definition of a positive, a negative, and a not determined. Not determined, of course, if you don't have enough cells, it's going to be not determined. But beyond that, so we said the positive control has to behave. That tells us our PBMC quality was fine. We also need a statistically significant result with a p-value less than or equal to 0.05. We decided that 2 times the negative control of the respective well was the cutoff. We also said that the minimum number of spots we need to see on the other spot is 40 per million. And so these were all things that came from our validation data, but I'm sure this is going to evolve as we get data from our patient cells. If the patient cell populations behave a little differently, we might have to change some of these conditions. But as of now, based on the validation data, this is what we called would be a positive. If any of these conditions are not met, it would be a negative. And if we don't have statistical significance, or if we just don't have enough PBMCs, we will just call it not determined. So this is sample analysis is ongoing. We've already analyzed some clinical trial patient samples with this assay. Everything's functioning well and as expected. So far, so good, keeping fingers crossed there. But I do believe that the assay is going to have to evolve over time to make it more elegant, I have to say, because it is the sample collection, the processing, everything can improve a little bit perhaps. Maybe our cut point can become more perfect in terms of calling the positives and negatives. But as we gather more experience, I'm sure we can improve on this assay. And we hope to be able to take all this data together and see how much value and how much meaning, how much value are we adding with this data and how meaningful is this? Is there correlation with PK? Is there correlation with efficacy? And see if we can maybe minimize the number of samples that we have to collect. And if we see we're collecting over 4 months, maybe it's always month 2 that's the highest, and then we can reduce it to just 1 sample per patient. And with that, I'll like to thank Jeremy. Thank you very much. It's been wonderful working together. I learned a lot, and I hope ProImmune benefited from us too. And Jim, the contracts for the clinical studies are no joke. These are studies that go over 3 to 4 years, and how do we structure the contracts for these sample analysis was an art in itself. Andrews is the scientist who's been PI for this project, and he's been doing all of the analysis. Divina has been responsible for data analysis and transfer of the data and the reports, which again for a clinical study is not simple. And my Celgene team and colleagues for all the discussions we've had. We've been growing along with the CAR-T world, and it's been fun. Thank you.
Thank you very much, Priya. Okay, questions?
Hi, very nice. I'm just curious about the peptide pool being 311 peptides. How was that achieved exactly? That sounds like a lot of peptides.
Yeah, Proimmune had reservations about it. There was just no way out of it. We couldn't make smaller pools. One of the ways we could have approached is to do some in vitro work to select peptides that were positive in vitro using a MAPS assay or something like that to make a smaller pool. But we were also trying to fulfill some regulatory requirements in this to say that we are indeed analyzing to capture any immunogenicity event. So sub-selecting a smaller pool we thought was not satisfying that requirement. So we do have 311 peptides. Beyond that, I'll let you guys address it.
We were, as I say, as Priya mentioned, we were a little bit skeptical as to whether that was going to be achievable with regards to background, particularly as being a challenge. But actually, we were pleasantly surprised. And we've also factored in, obviously, because when you plan for these sorts of assays, the number of the years that it's going on for, you also have to think about different batches of peptides that are going to be used in different arms of the trial as well. So we've also had to think about bridging studies for new batches of peptides. And again, that's where the positive control donors are very, very valuable. So really, it's just, I think there's a matter of luck, if I'm honest with you, a little bit with the fact that the quality of the peptides has been very good and we've had no issues at all with the background. That's been something that, if it shown up as being a problem, that's where we would have pointed it to, I think.
Yeah, we had to go with it and see how it worked. We've done 2 different programs now, this assay on, and both of them have worked pretty well. So, yeah.
Did you guys take special care about prepping a special purity or some sort
They
of-
were all 95% or greater purity. Yeah.
Thank you.
I'm going to go just to safety, sorry, just to get the order.
Thank you. So if you found any correlation with PK, efficacy, and any of the peptides or strong responses, say, in your ELISpot, would you consider modifying then your CAR-T?
Yes. Well, I don't know if we would go back and re-engineer these CAR-Ts, but I think right now we are still early. We're in the stage, okay, should we reinfuse a patient or not? If we have a patient who, we dose 10 patients, 8 of them are responding, 2 of them are not responding. Number one is, why are those 2 not responding? So there could be other reasons, but immunogenicity could be one of the reasons, so we can look at that. If they are not responding, will a reinfusion, second infusion help? So could we just reinfuse that patient to see if they are responding now, is one question. Because when we make a CAR-T prep, we have enough for 2 to 3 infusions at least. We infuse only at best a third of our prep. So if for some reason the patient doesn't respond, do we bring them back 10 days later and just give them a second infusion because it's already there, the cells are there on site? So that's a question we don't know what the answer is to that. And of course we want to look at if in some patients the first infusion gives a beautiful expansion. We see this great expansion and everything looks good. 6 months later the patient begins to relapse. And then if we give a second CAR-T infusion, the expansion is a lot smaller, the T cells are eliminated faster. So could there be a reason for that? Could immunogenicity be playing a part in that? Or is it something else? So there are a lot of questions we hope to answer before venturing into re-engineering. That's a different beast altogether.
Yeah, I was actually asking the same question, correlating the ELISpot data with the clinical outcome. Did you see anything you can correlate?
We don't have data. We have just one batch of data, only the first batch of samples, very small batch so far. So we don't have enough data to do those kinds of correlative studies and come to conclusions yet. But we have the assays now. It should be interesting.
Very nicely put together, thank you. I have a general question, like you use 250,000 cells in an assay and then report it as SFCs per million cells.
250,000 per round trip.
Yes, and you report it as SFCs per million cells, so you just multiply it by 4. But when you do your titration with lower number of cells, especially with the CEF pool and your positive controls, it doesn't titrate accordingly. It's like from 250,000, if you go to 100,000, 150, the response still remains the same.
Because it's not multiplied by 4, it's multiplied by, I think you get down to 100,000, multiplied by 10.
Okay.
So
Yeah.
it's proportional.
Yeah.
Okay.
Per million cells.
Okay,
SFCs per million.
I missed that.
Yeah.
So I was really interested in your PHA cutoff, because that seems really low to go down as low as 200 SFU per million cells and still considered viable. Was that due to the lympho?
I'll leave that. I had the same question. That's like always up there. Why did we choose 200? I'll leave it to
So
you.
it is quite a low number. If you look at the data, the actual PHA responses are actually typically a lot higher than that. I think it goes to what you were saying, Priya, that actually over time there could be something which, because a lot of the other factors are to do with the patient. So at the moment with the healthy validated controls, we could have set that way higher. But I think there's that question as we didn't want, I think we discussed that we didn't want to eliminate any of those patient samples on that basis automatically. Otherwise there may be an issue that none of those patients could then be analysed at all because we had that set. But
Yeah.
in reality it's a lot higher than that.
Okay.
Yeah, the thing is, when we were setting all this, we hadn't touched a single patient sample yet, and we were like, okay, we're setting all this, and we didn't want to eliminate every patient. Yeah.
Hey, Priya. I just wanted to understand how you're integrating these studies into the clinical trial per se. So you're going to be collecting this data for an individual patient's response prior to even the first infusion, or you will give the first infusion, then by the time they can potentially get the second infusion, these assays will be done and you'll have the data at hand?
Right now, no. So right now we are collecting one sample prior to first infusion. That's our baseline. And then we're collecting
the sample or doing the assay?
Collecting the sample.
Okay.
And then we're collecting 4 samples month 1, 2, 3, 4 post first infusion.
Okay.
And then it will all be analyzed. Whether we will have the data before a second infusion, that's not decided yet. So right now we are just in the gathering phase. We are not going to make any decisions based on this data yet. So
Okay.
we have to collect large enough volume of data to see if there is a correlation before we start making decisions based on the data.
So
right
that's
now
aspirational, basically.
Yes,
Yeah, gotcha.
aspirational. Right now it's
Yeah.
gathering. Irrespective of what the result is, the patient will get a second
Okay.
infusion if the physician feels they're ready for it. And then we look at correlative analysis to
So-
see if it makes...
But if this is your hope, then you think you have enough time from the first infusion to the second infusion to have this data in place?
Right now,
Not right now, but
later
eventually
on...
if you are going to do it routinely. How is it gonna
Yeah,
work?
so that's a bigger question, right? So if the physician wants to use one lymphodepletion and followed by 3 quick infusions or something, we won't have the time. If we think that immunogenicity is going to play a big part in the subsequent infusion and how well they characterize they are, then we will wait and time the second infusion such that we are able to generate data from the first infusion. So it depends on how valuable it is. If all of this doesn't correlate, this is all a wasted effort, but we won't know that until we get
No,
that.
I mean, but
Yeah.
it's good that you're trying to integrate it at least into your
Yeah,
clinical
yeah.
decision-making.
Yeah, I think over the next 2 years, it's going to be collect everything that we can and then look at it and then see if some things are completely not adding value, we'll drop it. If it's correlating, then we'll see, okay, how can we use this data? Should it be part of an exclusion criteria for a second infusion?
Last question with Andrea, and then we'll take a break.
Beautiful work. My question is about ADA. I suppose that what you're measuring is a post-infusion ADA. Is that correct?
Yes, post-infusion
So
ADA.
have you looked at, or have you thought about looking at basal reactivity?
Yes. So we do collect a pre-dose sample, pre-infusion sample, and then we collect post-infusion as well. And so far, all our pre-infusion samples are negative based on the data set. We have data worth 20 patients right now. That's how much we have. But all 20 were negative based on our cut point established using patient serum, but not CAR-T, not our study serum. So disease state matrix purchased from a commercial vendor was used to determine the cut point, and that cut point's holding up so far.
Okay, thank you.
Okay. Sorry, I know there are more questions, but feel free just to grab Priya afterwards, not too violently though. Thank you very much. We're going to have a quick break, grab a cup of tea, cup of coffee, and we're back in 10 minutes. So thank you very much. Thank you, Priya.
Thank you.